Okay, just a couple things to remind you of before we get started today. So remember that we're going to have quizzes every day this week. So Monday, Wednesday, Friday, there'll be a quiz on today's lecture posted by about noon today. Also remember that your practicals are this week. Now, I did get an email asking about a study guide for the practical exam. The study guide for your practical exam are the lab handouts that you've been getting all semester. So you guys should have access to those on your eCampus page. If not, get in touch with me or your lab instructor, and we'll make sure that you have all of the lab handouts that you need to study. Your tests, there's still a couple of people who haven't taken the last week's exam. I guess it was actually a week ago Friday. Time is running out if you want to make up the test. So please let me know ASAP, because I plan on handing test back 3 on one. Pardon me, test 3 I plan on handing back on Wednesday. So again, if you want to make up this exam, time's running out. You need to get in touch with me ASAP. And I think that's about it. So when we left off on Friday, we were talking about macrophages and what happens when they encounter anything foreign. So remember that macrophages are phagocytic cells. Okay, a phagocytic cell is a cell that does phagocytosis. Phagocytosis is engulfing, destroying, and digesting foreign invaders. Another thing that happens after a macrophage performs phagocytosis on a foreign invader is it becomes activated. Activated macrophages secrete chemicals that enhance the immune response. Some of those chemicals include, and this is the last thing we went over on Friday, chemotaxins. Chemotaxins are basically a chemical trail that other white blood cells can follow to the site of the infection to help get rid of that infection. Prostaglandins induce inflammation. A couple important things about inflammation. It's going to increase the capillary leakiness. This is going to help white blood cells like neutrophils to get out of the blood cells and to the site of the infection. White blood cells and serum leak into the tissues. Of course, this causes swelling, but this also enhances the immune response by allowing the circulating neutrophils to get out of the blood vessels and to the site of the infection. Endogenous pyrogen induces fever and drowsiness. Again, fever is going to raise the temperature. At higher temperatures, chemical reactions happen faster. So this is, again, this is going to enhance the immune response. Drowsiness is going to help you to conserve your energy so that there's energy to fight off the infection. Cytokines enhance function of other white blood cells. And again, there's a whole big catalog of different cytokines. Lots of different cytokines. These are unique to immune cells. They're the way that immune cells communicate and coordinate an immune response. So they enhance the functions of other white blood cells. Okay, then there's growth factors. So at the site of the infection, we're going to have damaged tissue. So growth factors are going to help to repair the damage done to the tissue by the pathogen. And also, some damage happens from the immune response as well. So that's going to help to repair that damage. Okay, so here's some pictures. So I mentioned that, in particular, not endogenous pyrogen, but prostaglandins make the capillaries a little leakier. So what happens is the cells that line the capillaries, okay, these are cells called the endothelial cells. the cell junctions are aren't as tight. It's when the cell junctions aren't as tight, this is going to allow cells to get out of the blood vessels by squeezing in between the endothelial cells that make up the lining of the blood vessel. So that's what we're seeing in A. This is a white blood cell squeezing out of a blood capillary by squeezing between the endothelial cells that line the blood vessel. On the bottom, so this is an example of phagocytosis. On the left-hand side, we have a macrophage. On the right-hand side, we have a yeast cell. And this macrophage is engulfing the yeast cell. It's doing phagocytosis. So it will completely engulf this yeast cell and then digest it. All right, so earlier when I mentioned natural immunity, one example of natural immunity is complement. And complement isn't just one thing. Complement is a set of about 20 different proteins produced by the liver, released into the blood, so complement is circulating in your blood. Complement becomes activated by pathogens. Okay, I see some of you still taking notes. I'll have a sip of coffee while you guys are finishing that up. Okay, so complement, it's activated by pathogens. Again, pathogens are these foreign invaders that cause disease. Complement performs several functions. So you may remember on Friday I referred to complement as the Swiss Army knife of the immune system. So it's got all these different things it can do. One thing is that it can activate other complement proteins. So some complement proteins become activated when they bind to the surface of the pathogen. Once they become activated, they activate other complement proteins. And then those activated proteins activate other proteins. And what happens is something called a cascade. So all of these proteins activating other proteins make for a whole lot of activated proteins. Okay, and these activated proteins can combat the infection in a couple of ways. Secondly, they can kill bacteria by assembling into a membrane attack complex, which perforates bacterial membranes. So when I talked about complement on Friday, I mentioned one of the things it can do is to punch holes in bacteria. Also, there are some types of viruses called envelope viruses that have a membrane around them. They can punch holes in those membranes too. Okay, so complement basically, when it gets activated by other complement molecules, it forms a pore in the bacterial membrane. And if enough of these pores form in the bacterial membranes, then the inside of the bacterium is going to spill out. And of course, same thing that had happened if somebody punched a bunch of holes in you, right? Your insides spill out and then you die. Same thing happens to the bacteria. Third, complement promotes inflammation. So again, inflammation is an important first line of defense. This is going to cause the blood vessels to become leaky. It's going to lead to swelling, redness, heat, all the things we associate with inflammation. Fourth, this is the word I was searching around for on Friday. I couldn't quite find. I don't know why I had a little brain fart on that. But they perform something called opsonization. Opsonization is coating of pathogens to promote phagocytosis. So when complement sticks to the surface of a pathogen, whether we're talking about a bacterium or a fungal cell or a virus, it makes that pathogen more attractive to phagocytic cells. So the phagocytic cells, they will engulf and digest coated or opsonized pathogens much more readily than uncoded ones. So the way this was explained to me by my microbiology professor when I was an undergraduate many years ago was he said complements like peanut butter. So you spread peanut butter on the pathogens, it makes them yummy for the phagocytes to come eat them. That's one, if it helps you to think of it that way, it's kind of true. It's kind of like complement makes the pathogens yummy. And that's called opsonization. All right, what we're looking at here, this is a transmission electron micrograph of a bacterial membrane that's being punctured by complement pores. So all these holes in the membrane, there are complement proteins that make these pores. And we can see all the pores. So this membrane is pretty extensively perforated. And again, once the membrane is compromised, like to this extent, the bacterial cell is going to die. So this is one of the ways that complement protects against pathogens. Okay, so next we're going to talk about, so previously we were talking all about natural immunity. Okay, the complement is a form of natural immunity. Phagocytosis is a form of natural immunity. And remember, that's just the first line of defense against pathogens. Natural immunity is great, but it's not specific and there's no memory. So while your natural immunity or your first line of defense is taking care of the pathogen, behind the scenes, your cellular immunity is ramping up. Your acquired immunity is starting to ramp up so that you can really attack that pathogen more specifically. So humoral immunity is one type of acquired immunity. So this is a type of an acquired immunity that defends against bacteria and viruses in body fluids. Okay, that's where the word humoral comes from. The word humor, I know today it refers, it's more like making jokes or something lighthearted. But the word humor actually comes from the word for body fluids. So back in the old days, before modern medicine, people thought that your body humors or your body fluids affected your mood. So when people would ask, what humor are you in? They were asking, what type of body, how are your body fluids affecting your mood, basically. So humoral immunity refers to immunity that uses the bodily fluids. Okay, so this is a type of acquired immunity that defends against bacteria and viruses in the body fluids. It involves protein antibodies in blood serum and lymphatic fluid. Antibodies, actually, let me go over to the whiteboard and let's write some definitions down. Because there's a couple terms in there you might not recognize. All right, I'm going to have to rearrange some stuff here. Okay, so first of all, antibodies. Antibodies are proteins of the immune system. Okay, and the thing that's special about antibodies is they're very specifically sticky for things on the surface of pathogens. Okay, so they specifically recognize and adhere to things on the surface of pathogens. We refer to these things on the surface of pathogens as antigens. So these are proteins of the immune system. Oops, sorry about that. Okay, so antibodies are proteins of the immune system that are specifically sticky to antigens. Okay, and antigens are surface determinants on pathogens. Okay, and your slide said these antibodies are present in your bodily fluid, so your blood and your lymphatic fluid. Now, you might not be too familiar about the lymphatic fluid. This is also referred to as lymph. So your body has another circulatory system. Other than your circulatory system, there's another system that circulates lymph, which is this fluid of the immune system. So there is some crossover. So your lymphatic fluid does drain your blood eventually. But the lymphatic system is separate from the circulatory system. And I might have mentioned this before. So whereas our heart pumps fluid through our circulatory system, there is no pump that gets stuff through our lymphatic system. The way that fluid moves through our lymphatic system is just from mechanical motion. So the mechanical motion of your breathing helps to move stuff through your lymphatic system. Exercise helps to move stuff through your lymphatic system. Okay, and this is a fluid of the immune system. And circulating, when stuff circulates through your lymphatic system, it enhances the immune response because your lymph nodes are part of your lymphatic system where immune responses are initiated. Okay, so it helps when stuff moves through your lymphatic system. I'm kind of getting up on a tangent here, but this is why people who do yoga or people who exercise a lot tend to get sick less, because the mechanical motion of exercising helps to circulate your lymph through your lymphatic system. That enhances your immune response. Okay, yoga, you have these really hard poses and a lot of deep breathing. Okay, that helps to move the lymph through your lymphatic system. And again, that enhances your immune response. That's going to be healthy. Okay, so sorry, I kind of got off on a tangent there, but lymph is the fluid that moves through your lymphatic system. Okay, so there's just some important definitions from that last slide. Let's go back to PowerPoint. Okay, so humoral immunity involves antibodies. Again, these are the proteins of the immune system that are very specifically sticky for antigens. Move through the blood and serum. So we know serum is the liquid portion of blood. Lymphatic fluid or lymph is the fluid that we find in our lymph ducts and our lymph nodes, throughout our lymphatic system. Antibodies produced by a type of white blood cell called a B lymphocyte or a B cell. These are the types of cells that actually make antibodies. Okay, I'm really not too concerned about teaching you guys about development of B cells. So don't worry too much about these first two bullet points. B cells develop from stem cells in the liver before birth or in bone marrow after birth. Okay, this first bullet point, don't worry too much about. Immature B cells migrate to and reside in secondary lymphocytes, spleen, tonsils, lymph nodes, etc. Okay, this is kind of an important point that the B cells go to these secondary lymphoid sites because this is where they will encounter antigen, and this is where an immune response is going to be initiated. Particularly in the lymph nodes. Tonsils are really just the lymph nodes. Of course, your spleen is a completely separate organ. At the secondary sites, B cells are activated upon encountering a specific antigen. Again, I define what an antigen is on the whiteboard. An antigen is a surface determinant on a pathogen. It can be a protein or it can be a polysaccharide. These surface determinants activate the B cells, and then the B cells will start producing antibodies that are specifically sticky for that antigen that they encountered. Okay, so I see some people still taking notes. I'll give you just a second to finish up. All right. Activated B cells become plasma cells that secrete antibodies that specifically bind to that antigen, like I mentioned before. There are 5 classes of antibodies: IgM, IgG, IgA, IgD, and IgE. Okay. Again, this is getting a little too far into the weeds. I'm not going to expect you to know all the different classes of antibodies. But you might be wondering, why do we have all these different classes of antibodies? IgM, during the immune response, IgM are the first type of antibodies that are made. So these are kind of like the first line of defense of the acquired immune system. IgG are the type of antibodies that we find circulating in blood. IgA is a type of antibody that we find on the mucosal surfaces of the body. IgD and IgE, these are involved in combating parasitic infections. And IgE, I think it's IgE in particular, if you have allergies, these antibodies are also involved in allergies as well. Okay, but again, I think this is a little too in-depth for a introductory biology course, so I'm not going to really be asking you about the different classes of antibodies. Okay, so here's a good picture. I was talking about the lymphatic system. So this is the lymphatic system. You can see the lymph ducts that connect all of these green dots. These green dots are lymph nodes. Okay, lymph nodes, if you kind of feel, right near your underarm, this is one part with lots of lymph nodes. If you kind of feel gently, you might feel little bumps under there. Those are actually your lymph nodes. So the lymphatic fluid or lymph travels through this system. The B cells migrate to these lymph nodes and the spleen as well. And these are the sites where these B cells encounter antigens. Once they encounter the antigens, they're going to become activated and they're going to start making antibodies that are specifically sticky against that antigen that they encounter. Okay, so this is a pretty good diagram of the lymphatic system. I know it's kind of hard to see, but you can see lymph ducts branching out all through the legs and the rest of the body. They're very thin. All right, so let's look at an antibody itself. So the structure of an antibody, it looks kind of like the letter Y. Up here, this is the ribbon diagram of an antibody. And you can see it vaguely looks like a Y. Down below in this diagram, you can see much more clearly how it looks like a Y. So there's 4 polypeptides or 4 separate protein chains that make up an antibody. We have 2 heavy chains and 2 light chains. So here's a heavy chain, here and here. These are the light chains, here and here. And the part of an antibody that's specifically sticky for the antigens is right here on the ends, like on the arms of the Y. So if you can imagine, if you're doing the YMCA dance, right where your hands are, if you're an antibody, that's the sticky part of the antibody. Okay, this is the part that's going to specifically bind to the antigen. Okay, and antibodies have 2. Okay, they've got 2 of these sticky parts, one on the end of each arm, and these bind exactly the same thing. Okay, so they have 2 identical binding sites. Again, that's going to recognize part of that antigen that they were activated by. Okay, so if, for example, the antigen that activated the plasma cell that made this antibody was on the surface of the bacterial cell, this part of the antibody is going to bind to that antigen. Okay, sometimes, well, this is a good example. This kind of looks like a coronavirus. Coronavirus has spike proteins, much like the spike proteins on this virus. Okay, this is not an unusual structure for a virus. Actually, influenza viruses have these spike proteins as well. And these spike proteins are usually pretty energetic, and they're a good target for antibodies. So in this example, we have an antibody that recognizes spike protein on a virus. Okay. Again, this isn't something that you're going to be tested on, but that's usually important because these spike proteins are usually how viruses invade cells. So if you can block those spike proteins with antibodies, then the virus won't be able to invade the cells. Okay, that's something called a neutralizing antibody when you have antibodies that do that. Okay, but again, that's just an example. All I want you to take away from this is that the antibodies are binding to these antigens on the surface of these pathogens. All right, so how do these sticky proteins, how do these antibodies actually neutralize or kill foreign invaders? There's 3 ways. Activation of complement. So certain types of antibodies, IgG in particular, fixes complement to the surface of the bacteria. So in other words, after antibodies bind to the antigens on the surface of a bacteria, then complement is going to bind to those antibodies and become activated. Okay. Now, this can initiate the complement cascade, and that's going to form those pores. Okay. In other words, it's going to punch holes in the bacteria, and again, kill the bacteria by punching a lot of holes in it. Okay. Antibodies neutralize foreign invaders by another way, by opsonization. So I already mentioned how opsonization works with complement. Okay, so complement's kind of like peanut butter. It gets spread on the surface of those pathogens, makes them yummy, makes them attractive for the phagocytes. Phagocytes come eat them. Okay. We also have a type of antibody called an opsonizing antibody. Okay. Opsonizing antibodies do the same thing that complement does. So they bind to the surface. Again, they're kind of like peanut butter, right? They bind to the surface. They make them yummy to the phagocytes, or they make them more attractive to the phagocytes. And that's going to increase the chances that they'll be engulfed and digested. Third way that antibodies can neutralize or eliminate foreign invaders is by agglutination. So remember that these antibodies have two binding sites. Okay, that's something that we call divalent. Okay, divalent means being able to bind to two things. Let me see if this is going to let me write today without going nuts. All right, well, it's letting me write, but not particularly neatly, but that's well, nothing different there. So try one time, then I'll go to the board. That's supposed to be a letter B. So D-I-V-A-L-E and T. So divalent, this means having two binding sites, or being able to bind to two things. Because antibodies are divalent, they can bind to antigens on two different bacteria or on two different viruses. When you have a lot of antibodies binding to two different things, it's going to cross-link them into a big complex. Okay, that's what's called agglutination. When these divalent antibodies cross-link these bacteria or viruses or whatever pathogen you have into this large insoluble complex. Okay. These large complexes are then eliminated from the liver. So they're going to travel through the bloodstream. Once they get to the liver, they're going to be eliminated from the liver. So that's another way that antibodies eliminate foreign invaders. Some activated B cells develop into long-lived memory B cells. Okay. And this is important. Actually, this is pretty timely because, of course, we're in the middle of a pandemic. We're all talking about getting vaccinated and getting long-term. So vaccination gives you long-term immunity. How does vaccination give you long-term immunity? All right. Now, again, I don't want to go beyond the scope of a general biology course by getting too deep into activation of these, how B cells are activated. But they do encounter antigen in the lymph nodes and the secondary lymphatic tissues, right? Once they encounter those antigens, they can activate it, and they're going to start making antibodies against those antigens. So specifically, a B cell is going to differentiate into a plasma cell, and a plasma cell just makes tons and tons and tons of antibodies. Okay? Also, while it's differentiating into a plasma cell, it undergoes something called clonal expansion. So it's going to divide a lot. So now we have lots and lots of these cells, all making a lot of antigen, all making a lot of antibodies against this one particular antigen, right? So as you can imagine, usually we're only sick for about 2 weeks or so. After about 2 weeks, once your acquired immunity has taken care of that infection, you don't need all these antibodies around anymore, right? And you don't need all these cells making antibodies around anymore. So most of those plasma cells are going to die off. However, a very few of them, a small fraction of them, are going to kind of go dormant and persist as what's called a memory B cell. Okay, these last a long time. They can last like a decade or even more. But what happens then, if your body encounters that same antigen again, then these memory B cells become activated. So the response is a lot quicker. You have a memory immune response rather than having to develop a new specific immune response. Okay, a specific immune response takes at least 7 days to start, and that's just the beginning of it. Okay, so it takes at least 7 days. That's a long time. If you have memory B cells against that same antigen, then boom, right away. So, and this is the whole point of getting vaccinated, right? So once you have these memory B cells that are circulating, when you encounter that same pathogen again, and with that same antigen on the surface, you have a much faster immune response. You know, so quickly you're not even going to get sick at this point. Okay, so again, this is going a little farther into the weeds than I want to get in a general biology course. But this is showing you how B cells become activated and then differentiate into plasma cells that make lots of antibody. This usually, what happens here is, trying to make this simple for you. So the B cell, when it encounters the antigen, of course, that's in the lymphatic tissues. It's the secondary lymphatic tissues, specifically, like places like the tonsils and the lymph nodes. That activates the B cell. However, we need a second signal in order for this B cell to differentiate into a plasma cell. That second signal comes from a helper T cell that has encountered the same antigen. Okay, so this is just kind of, again, you're not going to be responsible for some of the tests. I just want to kind of explain it to you, just so you understand. This is, how can I explain it? This is just to kind of prevent, to make sure that antibodies are being produced against a foreign antigen and not something that's part of your body. Okay, now we know if that could lead to an autoimmune disease, and we all know that autoimmune diseases are very bad. Right? Things like rheumatoid arthritis, Crohn's disease, multiple sclerosis. Okay, I could go on and on. All these things are the results of autoimmune diseases where your own immune system is attacking your body. By having a B cell activated by an antigen, and then a second signal from a T cell that recognizes the same antigen, then we prevent autoimmunity, which can be very bad. That's what's happening here. So at this point, so if the T cell recognizes the antigen on the B cell, it's going to secrete cytokines that activate the B cell. Okay, then the B cell is going to differentiate, it's going to undergo clonal expansion, where there's going to be a lot more of these. Okay, then it's going to differentiate into plasma cells. They're going to make lots and lots of antibody. It's going to attack the foreign invader. Again, then after the foreign invader is eliminated, most of these cells are going to die, and a very few are going to persist as memory B cells. And again, if that same pathogen is encountered again, it's going to be a very rapid response and a very rapid elimination of that foreign invader. Okay, so here again, some ways that antibodies can help to neutralize foreign invaders. On the left-hand side here, this is an example of agglutination. So look at, so we have, let's look at the individual antibodies here. So here's an antibody. So here's the antigen binding site here, antigen binding site, and we have the antibody here. So here's an antibody. Remember, these are divalent, meaning that they can bind to two things. So here's an antibody binding to two things. Here's an antibody binding to two things. Here's an antibody binding to two things. So by binding to two things like this, or being divalent, they make these large complexes. Okay, making these large complexes by binding to two things, this is called agglutination. So these complexes will circulate through the blood system. Once they get to the liver, these complexes can be cleared by the liver, and that gets rid of the threat from the foreign invader. Over here, we have opsonization. So the antibodies binding to the surface of this pathogen are opsonizing antibodies. So opsonizing antibodies make this opsonized antigen, pardon me, this opsonized pathogen more attractive to phagocytic cells like macrophages. So there are specific receptors on the surface of the macrophage that recognizes the antibodies. Okay, and so that's going to initiate phagocytosis, and these antibody-coated pathogens are going to be engulfed by the macrophage or neutrophil, and then digested, and that neutralizes the threat. Okay, so that's humoral immunity. Now remember, there's another type of acquired immunity called cellular immunity. Cellular immunity is a type of acquired immunity that defends against intracellular bacteria and viruses and cancer cells. Okay? So things like bacteria, some bacteria are just found in the body's fluids and aren't what are called intracellular pathogens. In other words, they don't invade the cells. Some types of bacteria, all of the viruses, they infect us by getting inside of our cells. Okay? Now, antibodies can take care of viruses as long as they're outside the cells, before they get into cells, right? But once they get into cells, there's nothing that the antibodies can do. Okay? Similarly, some bacteria are intracellular pathogens. You guys might be familiar with there's the bacterium that causes Lyme disease. Okay, that gets right inside of your cells. Now, antibodies can't do anything about that, so we need cellular immunity in order to take care of these pathogens that get inside of the cells. Okay, so this is the type of acquired immunity that defends against intracellular. That means pathogens that get inside your cells. Some bacteria are intracellular pathogens. All viruses are intracellular pathogens. Cancer cells aren't pathogens per se, but they are something bad that your immune system takes care of, and it's cellular immunity that takes care of cancer cells. Cellular immunity involves a type of white blood cell called a T lymphocyte or a T cell. Okay, so again, I'm not worried about you guys knowing how B cells or T cells develop. So don't worry too much about this first bullet point. T cells develop from stem cells in the liver before birth or in the bone marrow after birth. Immature T cells migrate first to the thymus to complete maturation, then to a secondary lymphoid site. Okay, so the secondary lymphoid sites are the same as what we saw for the B cells. So we're talking about tonsils, lymph nodes, spleen. Okay, so again, we see a diagram of the lymphatic system. So again, we can see the lymph ducts connecting the lymph nodes. The spleen is also a secondary lymphatic tissue. The thymus, this is like a little pyramid-shaped sort of gland that sits on top of your heart. This is where T cells are called T cells because the T in T cells stands for thymus. It's because this is where T cells mature. Again, I'm not overly concerned with you guys knowing about how exactly T cells mature. Over here, we have a cross section of a lymph node. Again, these little beads here are the lymph nodes. The lymph nodes are where B cells encounter antigen and also where B cells encounter T cells. Okay, so how does cellular immunity work? T cells produce T cell receptors, which are similar in structure to antibodies, but which always remain on the surface of a T cell. So if we look at the structure of an antibody, so we know, I've said before that an antibody resembles the letter Y. Now, if we took just one of the arms off of that Y, like right here, and then stuck that on the surface of a T cell, that's pretty much what a T cell receptor is. So just like antibodies can bind antigen, a T cell receptor can bind antigen too. Okay, but it's always connected to the surface of the T cell. T cells are activated upon encountering a specific antigen. Okay, again, the specific antigen is going to bind to the end of the T cell receptor, just like it would bind to one of the arms of an antibody. Helper T cells secrete cytokines, which enhance the function of other white blood cells. So in that example I showed you before, the T cell the helper T cell recognized the B cell, the antigen on the surface of the B cell, activated it so that it would differentiate into a plasma cell and make lots of antibodies. Okay, that's what a helper T cell does. Helper T cells really help B cells to differentiate into plasma cells and make lots of antibodies. Another type of T cell, we have a cytotoxic or a killer T cell. This binds to and induces the death of virus-infected cells, cancer cells, etc. Okay, so once pathogens get inside of cells, we really don't have a mechanism to get rid of the pathogen without killing the cell. So what happens is cytotoxic T cells or killer T cells recognize these infected cells, and what they'll do is they'll send them a chemical signal that'll basically, this chemical signal says kill yourself. Okay, and then that cell will undergo apoptosis. You may remember apoptosis from the development chapter. Apoptosis is programmed cell death. Okay, so the cell will just kind of fold up very neatly and die, and then that cell will be replaced, and what's left of that cell will be recycled. Okay, so here's an example of what happens in, again, you don't need to know this whole thing. That's kind of beyond the scope of a general biology course. But this is what happens in cellular immunity. So here's a macrophage that's recognizing a pathogen as foreign. So, oh, we're out of time? Okay, we'll finish this up on Wednesday. Okay, sorry, I didn't know we were out of time. If there's no questions from anybody on eCampus, I'll end the meeting here. Okay, again, remember, quiz today, I'll have that up by about noon. Also remember that you guys have your practical exams this week, and tests will be handing back on Wednesday. If you still need to make an exam, contact me right away. You're running out of time. Okay, that's it. I will see you guys Wednesday morning.